Hello everyone. In this video we are going to talk about the environment effect on the gene expression. Look at these two cute rabbits. So cute. What do you think the difference between these two? So you see that they have different colors around the nose, and also ears are a little bit different. These two have the black color, but this is no. What do you think? Do you think that they have the genetic differences? Pause the video and a little think about that. Okay, tell me what was the result of your thinking. Let's see. The environment of an organism impacts gene expression. For example, human hair and skin color are impacted by the exposure to sunlight and high temperatures. Okay, so what does that mean? This means that they have the same genetic information, but some environmental factors have changed that. Explain how the temperature will affect the expression of gene. What gene? Gene C. So what does it mean? There is a gene which is regulating the presence of the skin color in the form of black color or white color. If the gene which is controlling the fur pigmentation in Himalayan rabbits, both of them are Himalayan rabbit. This gene, when the temperature is higher than 15, and it is between like 15 to 25 degrees centigrade, gene C, you tell me, gene C, what happens to gene C? Gene C is inactive. Is not active. So when the gene C is not active or it's inactive, it doesn't make the color, it doesn't make the skin color to present as a black color, which means that it doesn't activate the pigmentation. So that's why there is no pigmentation, there is no black color happening here. But in the other side, or in the other hand, low temperatures, which happens at this rabbit, the gene C becomes active. So kind of gene C is in love with cold temperature. Even you can remember or memorize something like gene C loves cold. So gene C, who loves cold, means that in the colder temperature, it is becoming active and it starts to create the pigmentation. So that's why you can see these cute area of the nose and ears become a black color. So in low temperatures, gene C becomes active. In the rabbit's colder extremities like ears, nose, and feet. It's here as well. So their feet also becoming black and produces a black pigment. So what did you learn from that experiment? From that observation, that the environment will affect the gene expression. There is another example of the effect of the environment on the gene expression in the embryo. The environment of a cell can also impact gene expression. This is a complex area of genetics, but can be outlined as follows. So we are talking about the embryo and the effect of the environment. So Only a small number of genes are involved in determining body patterns during embryonic development. During embryonic development, there are very small amount of the genes that are involved in the body patterns. So, the expression of these genes is regulated by a group of molecules referred to as morphogens. So morphogens are not genes. Morphogens are molecules, are a group of molecules that can regulate and they can affect the gene expression. How does this happen? Morphogens diffuse across the surface of cells from a concentrated source. Therefore, different embryonic cells get different concentrations of morphogens. So depend on the concentrations of the morphogens in the environment, they can trigger, they can regulate the gene become active or the gene become silent or not active. So that's how they work. So it depends on the amount of the concentrations of these molecules around the cell. So morphogens regulate the production of the transcription factors in the cell. How do they do that? By the concentration around the cell, they actually control or affect the transcription factors. So transcription factors are something which is important for protein translation. So first of all, the transcription factors are having role in transcription, and then that transcription will lead at the end to the translation of that protein. So this is the beginning of the starting of that gene regulation. So these morphogens are controlling or are affecting the transcription factors. This results in the activation and inhibition of different genes in different cells. This in turn controls how long your fingers should be, where your nose is on your face, and other specifics about body structures. So wherever these morphogens are concentrated less or high, then that particular gene will be active or inactive, and that's the order to make it like become a nose, become a finger, how long does it become, so everything, so like that. Okay, let's watch this beautiful video regarding the gene expression.
So let's look, for example, just at 3 cells here. They're equivalent in potential. They could give rise to any region of the nervous system, but by chance they occupy different positions, and that position exposes them to different concentrations of this graded signaling molecule. So you can see 3 cells exposed to 3 different concentrations of this signaling factor. These factors interact with cells by binding to receptors on the cell surface. And then by virtue of the amount of signal, you activate receptors to different amounts. And then as a consequence of that, you initiate the activation of a first set of these transcription factors that are sitting in the cell waiting for the signal. So these factors are present, but they need to be activated either by addition of a phosphate group or clipping off a bit of the protein. And so as a function of the concentration of signal, you activate different levels of these transcription factors, the purple triangles shown here. And these are proteins then that once activated will seek out target DNA sequences with which to bind. And we see at this point that there's a difference between these 3 cells measured in the different concentration of transcription factor. So the cell on the left has a low concentration that is sufficient only to bind to gene A, whereas the cell on the right has higher concentration and can activate genes A, B, and C. And as a consequence, these cells begin to acquire different profiles of gene expression in the way that we saw. And these target genes, by and large, are themselves transcription factors. So this is a cascade of signal to first transcription factor to second transcription factor, that then these genes really start to mediate the process of establishing cell differences. And finally, through their secondary actions, cells acquire, in this case, different colors, but different.
So if I want to explain the video according to what we had in the previous slide, these signaling molecules that has been explained here is equal to that morphogenes. So if you go back to the embryonic slide, you will see that the morphogenes were responsible by the presence of the number of the morphogenes or the high concentration of the morphogenes. And here you will see that the more signaling molecules will create a strong signal here rather than the weak signal here. And if I go farther, then you will see that the strong signal creates the more transcription factors rather than the other one. And then the more transcription factors they can attach to more places and activate more genes. So more transcription factors because of the more signaling molecules, then they create the more they activate more genes. For example, here we have the A, B, and C. All the genes are activated. But here that the number of their signaling molecules were less, only 2 genes were activated, and here only 1 gene. So, and then finally, you will see that the target genes, which was all the 3 of them, A, B, and C, could create something that is not the same that has been produced at this level, at this stage. So presence, again, I go back from the beginning, presence of the signaling molecules, the higher concentration then will end with the presence of something more because of the activation of the more genes, which is totally different, or which is more than this cell and this cell.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.